Okay, finally, let's go. Welcome everyone to the 32nd session of STEM Quest 3.0. It's actually the final scheduled session, although we might have a couple special sessions between today and next week. You will be presenting some of the things that you worked on as participants. But it's a pleasure to have you here today. Today we have a very inspiring guest, Ariaye. He was a recent biology graduate at the University of Washington in Seattle, and he is a researcher in neonatal neuroscience and also a member of the Association of American Medical Colleges in the United States. Arian has done a lot of research, as I said, in neonatal neuroscience, also exploring things like gyrification, which he will talk about today in response to hypoxic-ischemic brain injury, and how that affects neurodevelopment in youth. So, Ariane, whenever you're ready, take it away.
Okay, awesome. You guys can hear me okay?
Yes.
Okay, wonderful. Thanks very much for the introduction. Amir Ali's introductions are always very generous, so it really hypes me up prior to beginning the presentation. So thank you. Alrighty, hey guys, I am Arian, and I will be talking about alterations in gyrification in response to hypoxic ischemic brain injury. And I will particularly be focusing on animal models today. Okay, next. Right. So as you're aware, STEMQuest has participants from all over the world, which is fascinating. And I wanted to share a little bit about my background, as I thought you may find it valuable. I was born in Afghanistan, and due to the raging war, my parents decided to flee from our home country. This was a huge sacrifice as they left everything behind. I was only 9 years old, and our destination was Turkey. From 2012 to 2019, we lived at a small state in the Black Sea area, extremely welcoming, genuine, and generous people. I spent majority of my childhood here, so I certainly consider both Afghanistan and Turkey as my home countries. However, thinking about the limited opportunities given to refugees, my family and I made another major decision: moving to the United States. So we knew that this would allow my parents, my siblings, and myself to truly witness the fruition of our efforts. That's what I consider also as one of the special aspects of living in the US. Even if you work for one extra minute, you know that you will be compensated, whether financially or in some other way. Your time and efforts are valued. Okay, so fast forward. I completed my last 2 years of high school at Kent Meridian, went to Greenwood Community College, and transferred to the University of Washington in Seattle, which I graduated a couple of months ago with my bachelor's in biology. At the University of Washington, I worked as a research assistant in the neonatal neuroscience lab. I had the pleasure of presenting my projects at various institutions and conferences, allowing me to share my learning experiences with curious minds, just like our group here. I aspire to become a physician. Here is Dr. O'Connor and myself. She's done so much for me. I can spend hours talking about the kind of doctor and human she is, but this is just a small way of expressing my appreciation to her. I am also a huge believer that every single individual has a story that needs to be heard by the rest of the world. I've been fortunate enough to be a voice for the ones that are unheard. And it is something that I aim to do until my last breath. So what you've heard so far were the visible part of my journey. Here's the real introduction, the introduction that can't be seen with naked eyes. Learning the Turkish and English language from zero, trying to adapt to changes in such a short time, carrying responsibilities as the oldest sibling, a son, a family, a community member, a student, an individual. I failed more than you can ever imagine. I felt shameful, worthless. I regretted some of my decisions. I always told that I am never enough. I doubted, put guilt on myself. fear that I may not see tomorrow coming. In fact, I'm still living with these emotions, but I've learned to remind myself that I have a family, instructors, mentors, and friends that care for me. Thanks to their support, thanks to their blessings, I turned these feelings into opportunities that led me to this journey of continuous learning. And if you ever feel any of these emotions, please know that you are not alone. By now, I can hear some of you saying that I didn't sign up to listen to the story of a random guy, and I totally agree with you guys. So we'll keep it there. Okay, so in case if anyone fell asleep, my topic was alterations in gyrification in response to hypoxic ischemic brain injury. It is quite mouthful, but we'll break down those terms along the way. Starting off with gyrification. So what is it and why do we care? Gyrification is the process of brain development where it leads to the formation of gyri, so the ridges, and sulci, the grooves. Gyrification significantly increases the surface area of the brain, which we can see on the right. This figure represents the MRI of a baby during gestation. Triangles are cerebral volume and circles are cortical surface. You can see as the brain volume increases, throughout gestation, the surface area of the brain also increases almost exponentially. More surface area corresponds to more space. And this allows a greater number of neurons to pack in our brain regions. And I'll expand on this sentence further in the next slides. These neurons eventually create vast number of connections, which gives us the ability to perform complex cognitive functions such as decision-making, problem-solving, language, attention, and much more. These are also some ways which we differentiate from animals and other species. Well, my cursor is kind of going an interesting way, but it's okay. Okay, so I did prepare another way to showcase gyrification. Imagine your brain as a balloon. You blow up the balloon, the volume increases, right? Just like our brain does as we grow up. You blow it up a little bit more, and if the brain isn't able to fold, so creating those gyri and sulci, it will pop, a very loud pop. And that is not good, right? So gyrification is important to stay alive. Expanding on what I've said earlier, so neurons packing in our brain regions, I want to take us quite early in time, during the development of embryonic neocortex. It is part of our brain consisted of 6 layers, and I'll focus on ventricular zone. Let me see the cursor right. Yeah, there it is, cursor. Ventricular zone and subventricular zones, because they contain progenitor and neural stem cells. Contain progenitor and neural stem cells. So, and what I mean by that is they're precursors, undifferentiated cells. They haven't developed specific structures or functions at such early stage. Around week 3 or 4 of embryonic development, neurogenesis, so birth of new neurons, began. Progenitor and neural stem cells mature, differentiating into specific cell types like neurons, oligodendrocytes, astrocytes, etc. And these are essential to perform the complex cognitive tasks I've mentioned. And I promise you this won't be the only time you will hear these terms throughout your STEM journey. Okay, so as we zoom into the embryonic neocortex, you will see radial glial cells, which play an important role in guiding migrating neurons during brain development. You can also see basal radial glial cells and HOPS+. They proliferate or rapidly increase in number, leading to the formation of gyri and sulci. Hear me out, guys. I always carry the same excitement as when I first learned about this stuff. The more I talk, the more I want to share. But okay. So before I go to my next slide, there's something really cool I want to share. HOPS+ is regulated by Sonic Hedgehog signaling, and you definitely heard it right. Sonic the Hedgehog himself. In fact, just like this. Check it out. Well, I don't think that's how it works, but at least what I imagine in my head. So SHH plays a major role during embryonic development. And as you can see on the right, going down, when it is inhibited or undergo mutation, it causes deformations. In fact, you guys can look it up online. There are really devastating conditions when SHH is lacking. It's pretty bad. So yeah, so all right, we learned a good bit about gyrification. Let's transition to the second part of our topic. Hypoxic-ischemic brain injury. It occurs when the brain receives insufficient oxygen and blood supply during childbirth. It increasing the risk of neonates developing, or newborn babies developing various neurological conditions later in life. It affects 1 to 3 babies in every 1,000 births globally, making it a leading cause of neonatal mortality and morbidity. The current standard of care is therapeutic hypothermia, so decreasing the body temperature, though it is only available in high resource settings. And unfortunately not neuroprotective in lower resource settings. You may have realized that I didn't mention term encephalopathy and said brain injury instead. That's because encephalopathy it's the term that we only use when referring to human babies. In our lab, to better understand the effects of hypoxic ischemic brain injury on gyrification, we utilized animal models. And I want to ask you guys, I want to ask everyone a question here. So what differences are you seeing when comparing the rat brain to other species? Either type it in the chat or unmute yourself. Again, no right or wrong answers. I really want to hear your thoughts. I want to be interactive. The size and the shape, okay. That's awesome. What else? I can repeat the question for sure. So what differences are you seeing when comparing the rat brain to the other species? Less gyrification, much smoother. Okay, wonderful. Now wait 10 more seconds. All right, no, those are wonderful responses, and you guys were totally right. So more neurons, much less lobes. Okay, yep. Again, those are wonderful thoughts. Rodents like mice and rats are lissencephalic, meaning they lack gyrification and have smooth brains, as mentioned also in the chat. Whereas ferrets, piglets, sheep, and primates are gyrencephalic. Like humans, they have gyrification. Along with some other conditions, this makes them translationally more relevant models. Additionally, what I've mentioned earlier about neocortex is still applicable here. And I want you guys to take a look at the difference between subventricular zones and subplate. Primates have more allocated space compared to mice for these special types playing an important role for brain development and gyrification. I'll also quickly show this. So the basal lamina, a thin but dense layer of extracellular matrix. Again, looking at the differences, gyrencephalic species have more progenitor radioglial cells and signaling mechanisms that leads to the formation of gyri and sulci. In our lab, we studied ferret and non-human primate models of hypoxic ischemic brain injury. And I'll first talk about the ferret model. So taking us to a paper, first author being Olivia Brandon. She was born and raised in the Neonatal Neuroscience Laboratory and currently a first-year medical student at the UW School of Medicine. She's also an individual that introduced and taught me all about the world of gyrification, as well as what it means to be a researcher. So shout out to her. Okay, so in our study, we utilized late preterm equivalent postnatal days, so P17 ferrets. In other words, 17 days old ferrets. We had two groups, control and injury. The injury group received lipopolysaccharide, triggering inflammatory response, and underwent bilateral carotid artery ligation. So this led to restriction in blood flow to the brain, mimicking the ischemia. Afterward, we placed them under a low oxygen environment, hypoxia, followed by high oxygen environment, hyperoxia, which is also the name of our injury model. And again, to reiterate, the control group didn't go through any of these procedures, and that's how we're able to make comparisons among two groups. HIH animals were randomized to receive saline vehicle, erythropoietin or EPO, therapeutic hypothermia, and uridine. At P42, so 42 days old, we took ex vivo MRIs. Each brain had 60 to 70 individual MRI scans. To understand and be able to quantify effects of injury, we used a metric called the Gyrification Index, or GI. This was done by taking the inner to outer hemispheric ratio, which is also illustrated on the right as well, right bottom. The inner trace follows the grooves of the sulci, and outer trace captures the outer circumference of brain without tracing the sulci. And if you're super curious on how actually did this GI tracings, those lines, feel free to scan this QR code, which will reveal the magic for you guys there. I'll wait for 5 seconds in case if anyone would like to scan the code. Okay, moving on. Okay, so to familiarize myself with the brain anatomy, I used a website called the Scalable Brain Atlas. You can see the slice number showing how, rostral, mid, or caudal the brain slices from, which you can also follow from this blue line here. Just some examples, this is slice number 11, 51, 91, 121, and 151. So it's really going throughout the brain. And, very useful. So I'll drop the link to the website at the end for you guys as well. All right, so moving on to results. These are quantile regression graphs showing a median GI. Recall when I mentioned that there were 60 to 70 individual brain scans. These graphs illustrate their GI values spanning throughout the brain. And our groups are indicated with different colors. So as you can see, the GI of female control and HIH were relatively similar, though we cannot say the same with male control and HIH groups. Sorry, I'm trying to use my proxy as well. Getting hard today. Furthermore, where GI reached its peak, you guys can see it's much closer to 1.3 in male HIH compared to female HIH and control, whereas male control is at around 1.2. So some box plot graphs, and a different way of showcasing our data. I have another question for you guys, and so sure we'll get there. So can someone tell me what is a p-value? And again, you're more than welcome to type it in the chat or on mute. Yep, significant findings, wonderful. Okay, great touch. We'll wait 5 more seconds. Okay, awesome. No, thanks very much for that response. That's you've truly captured what I was about to mention here. So p-value, also referred to as a null hypothesis, is the probability of our data coming together due to a random chance. The threshold is set as 0.05 or 5%. So if your p-value is lower than 0.05, your data is considered significant. So here we can see a significant change between the average GI of male control. Oh my God, these okay, male control and male HIH. Additionally, close to a significance threshold among peak GI of female control and HIH. So the main takeaways with these and previous graphs are, we found differences in GI, including sexually dimorphic effects, which I think are exciting. It gives us more reason to further investigate in such projects. And FYI, we've done so many other analyses, association of GI with brain volume, pathology score, behavioral tests like negative geotaxis, cliff aversion, which are reflex tests, as well as a regional GA analysis. For sake of time, I included just a couple of graphs, but I highly encourage you to read the paper at your own time, and please don't hesitate to reach out to me in case of any questions. All right, so moving on to the next paper, focusing on the non-human primate model of hypoxic ischemic brain injury. You may also hear me say NHP instead of non-human primate for short. Okay, going over methods again. So the study consisted of term-equivalent pigtailed macaques. And just like the ferret paper, we had the two groups, control and injury. The injury group underwent up to 20 minutes of umbilical cord occlusion, or UCO for short. In simpler terms, we clamped the umbilical cord, which restricted blood flow and oxygen, mimicking the hypoxic ischemic injury. So UCO animals were randomized to either therapeutic hypothermia plus EPO or no treatment, while non-UCO animals served as controls. MRIs were taken at 3, 4, and 7 days, as well as 6 months. We also did cerebral palsy assessments at similar timelines of the MRI. Last but not least, we obtained a GI matrix like before. Okay, so prior to moving on to results, just so we get a solid understanding of the broad picture, on the left, you're seeing the MRI of a ferret brain, coronal section, the non-human primate in the middle, and a human brain on the right. So as we are increasing in the complexity of species, the complexity of cortical gyrification also reflects that, right? So, I mean, just take a look at it, guys. So the gyri are much larger, sulci are much deeper. I mean, I think it's fascinating. How did we assess CP through behavioral testing? Yes, it's a wonderful question. So to assess CP, we had professionals who has done these jobs in the past, whether neonatologists or other related individuals, and they had a scaling, a kind of a scale score, which also kind of measured the severity of CP as well too. So that's how we measured, relying on the professionals who has done this job in the past. Okay, so moving on to results here, but a fun fact actually, the study itself was done back in 2014, so almost a decade and a little longer ago, but no one conducted GI measurements back then. I kind of took on this project doing a secondary analysis, specifically concentrating on gyrification. Therefore, the figures I'll be sharing aren't published anywhere. So you guys are basically VIPs, as of course as you are. So I think it's safe to say that our lab will be the first group of people to share these findings in the near future. So another quantile regression graph showing median GI. So they're here relatively similar. Scatter plot on the right, x-axis being the treatment groups and y-axis the average GI, and we did not see any significant changes. So moving on to peak GI. So on the left, we have all three of our treatment groups, whereas on the right, we have we combined the UCO, so the injury groups. And because non-human primate studies are underpowered, so in other words, have smaller sample size, it's quite common to combine groups of interest. So in order to make certain adjustments, though we did not see any significant changes here either. All righty, so before I show you guys more figures, does anyone know what this is? Anyone knows what this is? I know it's a funny picture, but it's homunculus. You've got it exactly right. That's exactly what it is, and specifically, it is cortical homunculus. And homunculus actually stands for little human in Latin. It's basically a buckled map of the human body projected onto the cerebral cortex, indicating that each gyri associates with a certain function. And it's an important concept as we dive into what we have here, regional analysis. So So as kind of the name reveals, we're focusing on a particular area of the brain, right? So regional. And we examined what gyrus associates with motor functions, as well as where it kind of begins and ends. So, that's how we came up with the graph on the left. The motor GI of control and UCO animals are, relatively similar, though we do see differences in patterns around slice 20 to 25. On the right, just like before, we're seeing scatter plot graphs, and we did not have significant change among our groups. The other area we focused on was somatosensory, so relating to sensing touch, temperature, pain, etc. So control and UCO looks relatively similar again, but there are differences in pattern between the slice 50 and 60. So and we did not see significant change with scatter plot graphs here. Alrighty, so here we separated our groups by sex. Top two graph has all three treatment groups, and bottom two has the UCO groups combined. Looking at our top graphs, their median GIs are relatively similar. But I want you guys to look at the control group, so the blue line. With males, they reach their peak around slice 60, whereas with females, around slice 44. So it's, it is interesting, isn't it? So looking at bottom two, controls remain the same, and with UCO, you can see the peak of females, female UCOs are tiny a bit closer to 2.8 compared to male UCOs. Again, I hope I'm using my cursor, kind of the screen shifts the cursor, so I'm trying my best to go as slow as possible there. Okay, so this time we combine all treatment groups, control UCO with HT placebo and UCO with no treatment, and separated them by sex. So same drill, average, peak, motor, and somatosensory GIs. And we did observe the average GI of females being significantly higher than males, top left graph. I'm curious, what do you think may have led to a significant change? And do you think we can truly conclude that females have higher GI than males? Again, more than welcome to speak or type it on the chat. And really want to emphasize again, there's no right or wrong answers. I really want to hear thoughts. I really appreciate any contribution. Again, take your time, not a problem. We'll wait around 30 more seconds or so. I know it may be scary to share your thoughts, especially in these graphs, but please don't worry about being wrong or right. But again, there's no right or wrong answer, I promise you guys. Amiral, may you please read the entire sentence? I'm not kind of restricted on the screen there. Really appreciate that.
Yes, sure. Yumna said, I don't think we can necessarily conclude just from this study that it would be the same in humans. Okay, wonderful. That's a wonderful thought. And again, that's certainly a potential thought there. What you mentioned about the difference there, but also imagine at the beginning I mentioned that we combined all groups, right? So UCO with HT placebo control and UCO with no treatment. And again, there may have been some contribution in that line, but also thinking about anatomies, right? Anatomies of non-human primates as well as humans, and there are hormonal effects that may also contribute into this. Again, so there are various reasons. We do not know why, but that's why I said there's no right or wrong answer. Again, it could be really anything, but that's just something exciting there. Amir, were there any other thoughts before I move on? No, other than Faith agreeing with it. Okay, awesome, yeah. Awesome. Thanks very much. Okay, so as you guys may recall, I mentioned that we also did cerebral palsy assessments.
Here, we separated our non-human primates as the ones diagnosed with CP versus no CP. Median GIs across the brain on the left, animals with CP, kind of reached a higher peak. Motor strip in the middle and somatosensory on the right. And, you guys can see patterns are certainly different. So, just some thoughts to keep in mind here. In the scatter plots here, so again, same deal as before, separated our animals diagnosed with CP versus no CP, and we did not observe a significant change. I know I went through a good number of graphs, but I hope they kind of helped you to get a better understanding for the various questions you can ask, and as well as the stats you can run. So hopefully that provided you some more thoughts as well there. We actually ran the same set of statistical analysis with MRIs taken at 7 days, but I decided to not go over them all. Instead, I just want to give you guys kind of a little glimpse. And this is what we call as trajectory GI, examining how GI has changed from 7 days to 6 months. And as you guys can see, GI seems to decrease in non-human primates over time. I'll leave it here, but think about the questions we can investigate into after coming across such data. GI decreasing over time from 7 days to 6 months. In fact, a significant decrease with average GIs as well as somatosensory. But again, just some thoughts for you guys to think about. And again, I hope this doesn't become a nightmare as you sleep, but some positive thoughts or rather healthy thoughts. Okay. So in conclusion, gyrification is an essential concept to explore the potential shared developmental processes between gyrified animals, so ferrets and non-human primates, and the neonates or newborn babies. As gyrification continues after birth, early detection of changes due to hypoxic-ischemic brain injury can expand the scope of providers to better assist neonates diagnosed with HIE. And this includes detecting possible long-term neurodevelopmental impairments and developing preventative interventions accordingly. So, what's next? What are our next steps in this project? Well, majority of the statistical tests that we ran weren't significant. Though we saw differences in patterns at particular sections. So therefore, we'll be conducting gyral, sulcal, and aerial analyses, examining each gyrus one by one. We'll certainly take some time, but I find so much joy throughout the process. Another thing is, can early GI matrix, so 7 days, provide us some clues on the potential condition of the baby at 6 months, right? Is there a GI value that may associate with injury, right? Another thing is that we also have cytokine and chemokine samples taken at multiple time intervals. Some examples include interleukin-6 or IL-6, TNF-alpha, IL-1 beta. Basically, what there is they're signaling proteins. They allow us to have a better understanding for the severity of the injury and response to therapy. Are there any associations between these biomarkers and changes in GI, right? So various paths how we can follow. So I also have some optional self activities which you guys are more than welcome to do in your spare time. You can write about a condition that occurs when sonic hedgehog signaling lacks during brain development. Or using the scalable brain atlas, you're more than welcome to just poke it in and select the gyrus that looks interesting to you, and write some, just some questions for you guys to think about. Where in the brain that the gyrus generally resides? So what is it, and what is its functions? And I have some tips. So I'll share a screenshot of this on the WhatsApp group as well. So don't worry about kind of having everything down in here. So I'll definitely make sure to have that ready for you guys. So I want to wrap up with some kind of lessons I've learned throughout my journey that helped me to be here today doing this presentation. So I gotta tell you guys, I didn't depend on the compliment of others. Okay, so I know I achieved things or great things, but didn't necessarily live with the pressure of reaching something greater, so it kind of becomes worthy to share my learning experiences with others, they're in person or in the virtual world. I've done everything because I wanted, right? Because I found a value behind it, because it allowed me to become a better version of myself. Guys, we gotta feel the discomfort, right? The stress, the pressure, that will always be there, right? There will be days where we'll feel like there's no tomorrow. And I promise you guys that the decisions we take during these moments are truly what makes the difference that we would like to see, even if it's a tiny bit. Time. We gotta give ourselves time to adapt, right? I mean, just consider my story as one example, and I know we have so many people here. Imagine about your story, right? I mean, just think about it. You gotta give yourself your time to adapt, whether it's from transitioning from middle school to high school to college, or from one job to another, from one country to another, from one situation to another. You gotta give yourself some time. And I think it's important to remember our whys, and it could be anything. And I won't go much in depth, but I want you guys to ask yourself this question, right? What happens when you don't give up? And just imagine everything that you can do when you put that thought into your mind and actually apply it. And it's funny, but I promise you guys, it feels better than winning 5 matches in a row in Clash Royale. So I promise you guys, it feels way better than that. All right, so I cannot end this presentation without expressing my gratitude to the NNL Amelia. That's kind of our crew. No matter how interesting your research may be, the research, the work that you do, again, that's my personal thought. But as if you do not have people that truly supports you and want you to learn, want you to make mistakes, want you to learn from them, I don't really think that's really the place to be in. So again, these are the people that helped me to just get a better understanding of the research world, get a better understanding of what it means to be a researcher, get a better understanding of what it means to be a human that works towards making the lives of someone, or I should say tiny lives, better. And again, just so much appreciation to our team. I can stay as long as with the slide. It really brings so much memories and emotions here. So shout out to every single person on our team. And I thank you guys. Thank you guys for separating your time. Thank you guys for listening to me. And I want to thank the YSI community, YSI staff, as well as their members for having me and providing me this opportunity to share a little bit more about the world of gyrification and just my experiences. All right, and if you're guys are interested in staying in touch, my contact information are here. More than welcome to scan the QR code. You'll have my email, LinkedIn, Instagram, whichever you feel comfortable. More than welcome to connect and have further conversations. So once again, thanks for having me, and I'm happy to take on any questions or thoughts you guys may have.
Amazing presentation, Arian. Thank you so much for that. All that information about gyrification and the research that you did in neonatal neuroscience, and as well as the advice that you gave at the end. I think that's something that we'll take to heart and we'll cherish to become better people and improve without needing the validation of others. So thank you so much for that. And to the participants, if you have questions, feel free to just unmute yourself and ask, or type it in the chat.
Okay. Good. Thanks, Yamna. I hope I'm pronouncing your name correctly. Going back to the slide before the summary, could you decrease? Oh, so could the decrease in GI be due to more specialized use in certain areas compared to others as you grow older? So if I understood your question correctly, I believe you're asking whether a decrease in GI could be like as we grow up, whether certain areas like shrink or extend or change in structure or function. Did I understand that correctly?
Sorry, I think it'll be easier if I say it. So you said, I think the results from P7 to 6 months.
Correct.
So I know as you grow older, depending on different lifestyle factors, environment, and other factors as well, there is generally a kind of decrease in the like folds that you have in your brain. It's just part of normal aging. But I was just surprised about seeing such a big decrease, I suppose, in such a young age. Whereas when you're kids or children in general, they have a more, they kind of use their brain a lot more when because they're more exploratory than like our current age. So I would just assume that they would have like that it wouldn't have such a big decrease from P7 to 6 months.
Mhm. That's a wonderful observation. You know, I'm so glad you mentioned, you know, that like what you mentioned could be one of the many reasons that we have seen that decrease in non-human primates. And again, our thought is that because non-human primates are one of the most translationally relevant models, you know, it certainly gives us some thoughts into what it may look like also in human brains as well. But of course, no promises. We're not necessarily sure how it may look like um, um, in human brains. Though, you know, kind of adding on to what you mentioned, um, it could be, you know, also for instance, like especially in the ferret paper, as you get through the statistical analysis, you will see that we've done something called adjusting. And what adjusting means in statistical analysis is that you adjust for, let's say, for instance, like volume of the brain. Perhaps some brains may be larger than the other, as you mentioned, right? Or their perhaps weight, or you know, things along those lines. So there are certainly various factors that goes into these differences that we're seeing. And again, through our work, we just kind of had a better understanding of what potentially may be contributing to these decisions. But no, I truly appreciate your the observation there. That was just very well said. Very well said. This impaired neurodevelopment increases the risk of increased brain electrical activity, hyperactivity, and brain imbalance. You know, another wonderful question. So I think I said it quietly for myself. So does impaired neurodevelopment increase the risk of increased brain electrical activity, hyperactivity, and brain imbalance? You know, it's a wonderful question. I wish I had a definitive answer, but it is very possible. It is very possible. You know, as we are seeing kind of effective injury or some sort of impairment, right? There are various neurodevelopmental risks, or I should say, the risk of developing neurodevelopmental conditions later on certainly increases. And you know, increased brain electrical activity, hyperactivity, brain imbalance. You know, those two terms at the beginning. Increased brain electrical activity. It may be good and perhaps maybe not the best, depending on the situation. Right? There are various conditions that may change depending on what you're thinking of. Hyperactivity, again, for some it's great and for some it's not. So you know, those are some great thoughts. But you know, as you've kind of concluded with brain imbalance, certainly you know, one small change can contribute into bigger change that perhaps we're not able to observe from an outside world, but we may, you know, our body may react to it in ways that we have not realized that. And, you know, that's kind of the goal, right? So, you know, is there a possibility that we may be able to detect these potential changes early on and see whether those changes lead to any sort of impairment later on? Again, those are kind of the ultimate questions that we hope to deliver, and you know, step by step, or one day at a time, as mentioned at the very beginning of our talk. You know, we're kind of getting. I know we'll get closer and closer. But again, really wonderful thoughts and things to kind of keep in mind there as well. I truly appreciate your participation. It's really awesome. I'm glad, you know, it gives you guys some sparks into learning more about gyrification. So again, yeah, really more than happy to take on more questions. You know, whether it's research related, you know, any other part of the presentation, happy to take on. And again, more than welcome to unmute or chat. Hopefully, yeah, I really love this environment of kind of comfort and ways that we're able to share our learning experiences without the sense of judgment. It's really an open space for everybody.
If you have other questions, you can also, I mean, that come up later. You can ask Arian in the STEMQuest group chat on WhatsApp. Arian, sorry to give you more homework, but
no,
I love answering questions. So if
I
any of you have questions, just feel free to type them in the group chat.
And no, I do not mind at all. I'll share the QR code as well as the activity for you guys on WhatsApp as well too. But yeah, no, pleasure is truly mine.
Fantastic, thank you. Okay, if we don't have any other questions at the moment, again, feel free to ask them later in the group chats. But thank you so much to everyone for attending this final scheduled session of STEMQuest 3.0. Again, we might have a couple special sessions later this week or next week. So stay tuned for that. Otherwise, thank you so much for attending all the sessions up until now. And a huge thank you to Arian for teaching us about gyrification and the amazing research that he's done at the University of Washington. You're truly inspiring, and I love all the content you post as well on social media. For those of you who haven't seen yet, he has an Instagram account where he posts about a ton of different opportunities that you can get involved in research across a variety of institutions in the US and beyond. So make sure to follow him and stay up to date with all the resources that he provides. I've discovered a lot myself from his page. So thank you so much for providing all these opportunities and sharing about them.
Of course. No, thank you. No, I'm really, you're again very kind. I appreciate the shout out. And I think that's really the goal, right? If we're able to provide some sort of information, even if it means in the smallest way possible, to one person, that's really what it takes. So one person at a time, one day at a time, and one opportunity at a time. So no, thank you.
For sure, and you embody that very wonderfully. So keep providing these resources, sharing about them, so we can learn and maybe apply to become researchers in neonatal neuroscience or related fields as well. And thank you so much for all that you do and for presenting today for us.
Awesome. Thank you, guys.
Thank you so much. All right. Have a great day, everyone. Take care. See you soon.
Bye, guys.